Assalamualaikum and good day. So this topic is basically solely on mass spectrometry. So I have a different slides than the one that you have. Probably because this is a new topic that I incorporated about 2 years ago, but this is actually a crucial element and the core technology being used to sequence protein. So let's get to know what mass spectrometry is. So it is a technique to produce charged molecular species. So the keyword here is charge. So separation of charged molecule, okay, is done via magnetic fields, because without which then you wouldn't get the charged molecule. And this is, of course, based on mass to charge ratio, or M over Z. It measures, okay, either 1 plus, okay, 2 plus, or 3 plus charge state. Now, what is the function of MS? Of course, to measure the mass, and also it will give you some information on the chemical structure if you were to proceed it further. But more or less, again, measures the mass and more or less kind of like for identification of your proteins and peptide. So these are some of the applications of MS. So you could basically do some identification, verification, or quantitation of either metabolites, proteins, drug, peptides, synthetic organic chemicals, or polymers. So it's endless. Not only MS is subjected to only protein, but some other compounds as well, including metabolites. Let's have a look at the basic principle of MS. So only three basic principles: ionization, separation, and mass analysis, followed by detection and generation of mass spectrum. So this is done by using fingerprinting approach. Okay, so remember our PMF, okay, peptide mass fingerprinting. So you can either have two approach, okay, either top down or bottom up. So in a top down approach, you separate and analyze intact proteins and directly analyze the intact protein without proteolytic digestion, which a lot of time people don't really prefer, but now it's becoming a favorite, especially in peptidomics. For the bottom up, this is the most common fingerprinting approach. Okay, separate and analyze digested protein, and of course, digested means that the protein needs to be cleaved or digested first, and of course, it analyzes peptide mixture by mass spectrometry. So, what are the components of MS? So basically, these are the components of MS. Okay, so you have to have an ion source, you have to have a mass analyzer, you have to have a detector, and you have to have a signal processing, okay, to get your output or to get your data out. So along the way, okay, you have a sample inlet, okay, that will go through your ion source, okay, and basically you have an instrument control data processing, okay, that will more or less, okay, conduct all these components in the machine. Now, the components basically involve sample introduction through the inlet, sample ionization, sample transfer to high vacuum region, and of course this is not easily seen or not viewable by naked eye. It's actually inside the machine. Ion mass to charge filtering, also inside the machine. Ion detection, again we have a detector to detect it, and data acquisition and further analysis. So this is kind of like a rough schematic diagram of MS experimental workflow, whereby you basically have your protein. So don't be a bit confused, okay, of the flow. So this is where it starts, okay, from your protein. Okay, you have your proteolytic peptides, which means that it have been digested or cleaved with proteases. You basically, okay, let it go through MS. Okay, so this is a typical or classic case of MS using HPLC or liquid chromatography. And then, okay, once it has been ionized, okay, by different sources, for example, ESI or either MALDI, regardless of the machine, the types, which we will see later on. So you can get, okay, all the masses, okay, and the different charge of protein. And you can either get, okay, just, okay, one MS spectrum, okay, or tandem mass spectrometry output. So there is a difference, okay, between MS and also MS, or what we refer to as tandem MS. It will be in a later slide, so hang on. So in the workflow, first and foremost, again, what you need to do is that you need to, of course, okay, it starts with the protein. So you need to make sure that your protein is pure, and you need to more or less make sure that if you were to fractionate or you have a specific target protein, you can fractionate it further. And this could be basically detected or visualized further by gel electrophoresis. Then you proceed with cleavage or digestion. So most of the time for MS, the protease used is trypsin. There are other proteases to be used, but a lot of time the machine the MS machine usually comes with a certain standard. So typical standard is trypsin, but if you were to add in some more money, okay, you can add in chymotrypsin, pepsin, and also derivatives of trypsin and chymotrypsin for the identification. So once your protein have been digested, you have peptides, mixture of peptides, then you can start injecting into the machine and have it analyzed again to produce the MS or 10-MS spectrum. Now, let's go into the ionization source. So there are three phases, okay, for ionization source, either gas phase, solution phase, or solid phase. So they basically would determine the types of MS to be used, okay, but your protein, okay, could go through either one of them. It actually depends on your preference and how willing you are to provide for the cost. Okay? So for gas phase, we have electron ionization, chemical ionization, or photoionization, which is quite common, okay, using GCMS. We have solution phase using the LCMS, liquid chromatography, by electrospray, or atmospheric pressure, or another atmospheric pressure on the PI and atmospheric pressure on the CI, on the chemical ionization, okay, and also photonization. So in the solid phase, okay, it's actually done, okay, by using MALDI, metric assisted laser desorption, or plasma desorption, or fast atom bombardment. Now for deionization, what happens in the process is that large molecules is broken into random fragments. So the keyword is random fragments. So it is a non-selective fragmentation, and hence it will actually not be easy to interpret. So that's why a lot of verification needs to be done. So the properties of ionizer in different types of MS, okay, basically there are lots of types of MS, okay, but the properties of ionizer usually have to have high ionization efficiency, stable ion beam, minimum background ion current, and less cross-contamination in successive samples, because you do not want your less samples, okay, to be contaminated with previous samples. So from ionization, okay, we move to the mass analyzer itself, the core. So in a mass analyzer, it disperses ions based on mass to charge ratio. So it focuses on all mass resolved ions at one single focal point. So, and of course, this is done under high vacuum. So different types of mass analyzer are available for you. Okay? So this basically determines, okay, the machine, okay, that you use, or the machines available being sold or when you ask for service. So they are ion trap, quadrupole, ion cyclotron resonance, orbitrap, magnetic sector, and time of flight TOF. So you may recognize it as MALDI-TOF. Now, what are the specifications of mass analyzer? You have to have a mass range, okay, which means that, okay, it would have a maximum allowable mass to charge ratio amenable to analysis. Provide resolution, okay, to separate two neighboring mass ions. Adaptability to different kinds of sample, which also basically more or less, okay, related to sensitivity. Okay? So next is efficiency, transmission multiplied by the duty cycle, because you can have different kinds of a gradient to be used inside the machine. Mass accuracy, so this is crucial, okay, measurement of mass from the actual mass, okay, there is basically kind of like a comparison that we will do. And speed of the spectra acquisition per unit time. So when you have one kind of like spectrum, okay, we call it as a spectrum, but when you have lots of spectrum, we call it as a spectra. So sensitivity, okay, which is, like I said, correlated to adaptability, is the minimum concentration of a compound that the instrument can detect. It actually comes back, okay, to the column being used as well, because different types of mass analyzer, okay, it depends on the type of column being used, okay, the sensitivity. And of course, okay, the last part is on the mass stability. So let's have a look at some example of the mass analyzer. We'll start off with a quadrupole, okay, which is the most common. So as the name says, quad, okay, not the quad muscle, okay, but quad means that four. Okay, so you basically, okay, have four poles, as you can see over here, but it's actually in the machine, so you couldn't see it with your naked eyes. So it has four parallel metal rods, lets one mass pass through at a time, not all of them, okay, which means that they have been screened, okay, during ionization, then only one of them, okay, will be passing through one at a time. So it could scan through all masses or sit at one fixed mass. So if you have one target mass, okay, you just fix it, okay. But okay, if you want to do peptide mass fingerprint, okay, so you can scan through all the masses, okay. So it will be scanned by the detector. Now, quadrupoles have variable ion transmission modes, okay, whether in mass scanning mode, okay, which means that you want to detect each and every one, or single mass transmission mode, okay, so you have a specific mass that you want to detect or target protein. So next, again, we will see time of flight. Mass analyzer are usually correlated with MALDI. So ions are formed in pulses. Okay? So basically, okay, this is what is happened inside the machine. Okay, time of flight tube. So the drift region, okay, is usually fill free. Okay, which means that from the ionization up until here, okay, is actually fill free. You don't really need high vacuum. Okay? So measures the time. Okay, that is why it's time of flight for ions to reach the detector. Okay. So this is what is referred to as time of flight. Don't ask me why it's flight. So small ions reach the detector before the larger ones. This is a classic case, okay, of a lot of stuff, okay, in physics as well, okay, like in electrophoresis. So small ones, okay, will go further down, okay, or will get everywhere faster because of the smaller range and probably, okay, because of its lightness. Now, the next one is the ion trap mass analyzer. So ion trap, okay, usually you will have four components as well, or four sample plate. Yeah, as you can see, this is kind of like this, 1, 2, 3, 4. Oops. And again, it is mostly associated with quadrupole. Okay? So usually we have a quadrupole ion trap mass analyzer. So similar application to quadrupole. So ions again will be detected again once it goes through the electron gun. All right. Now, those are some of the examples. Like I said, again, we're not going to go through all of them. Those three are the most common. Sorry, three or four. Okay, they are the most common and widely used. Okay, so we will move to the detector because if we were to cover all, basically, well, we can have one whole course on MS alone. Okay, but we don't have time for that. So in the detector, okay, what we have, okay, over here. So this is the overall flow, okay, of MNS and MS. Okay, so the detector is here. Okay, after the mass analyzer. So detector could be in the form of micro-channel plate or electron multiplier or hybrid with photomultiplier. It depends on the kinds of machine. And of course, again, the price would be higher as you go down the choices. Now, these are some of the examples. Okay, so with microchannel plate, okay, so ions, okay, could be detected. So the microchannel plates, okay, basically, okay, looks like this, okay, and the plates, okay, is actually here. It's actually just kind of like one small thing, okay, but of course, okay, this is a blow up, so you can actually see that it's kind of like thick, but it's actually very thin. Okay, so this is how the plates look like. Okay, so it has more or less a pore in between them. Okay, so this is where, okay, electrons are being detected, the parent and also the daughter ions that basically is bounced through the channel plate. And in the data system, okay, which follows the detector, okay, usually we have a PC whereby, okay, we can gather all the detected ions, okay, and sometimes, okay, for a lab or for a service lab, okay, usually they have stations. Stations mean that, okay, they basically are equipped, okay, with different kind of analyzer and also analysis tools, okay, coupled with it. So if you only have PC, usually it's just a machine plus a PC, but if you have a station, usually you have a few more other PCs, okay, just for the analysis. Now, what do you have, okay, when you send your samples, okay, for analysis, okay? So it shows, okay, the results of a spectrum or spectra. So as you can see over here, so peaks, in other words. So this is an example of Multitoph, okay? So this is a spectrum of IgG, okay? So basically, okay, we have, okay, different charges, okay? So 1 charge, 2 charge, and 3 charge ions, okay, being detected. So how do you know which one is which? There is a way to do that, but of course you have to have a standard, okay, to compare it with. It also shows, okay, some of the ESI spectrum of trypsinogen, for example, and this is done using LCMS. So the other one, previous one was done using MALDI, this one using LCMS. So you also have different kinds of spectra. Now, how do mass spectrometers get their names? So basically, it depends again on the types of ion source, types of mass analyzers as well. So for example, okay, if you have, okay, And usually when we talk about mass spectrometer, okay, usually they will ask, okay, so which mass spectrometer, okay, do you want to analyze your sample with, okay? So you either have to state, okay, the types of ion source or the types of mass analyzer of your preference. So for types of ion source, we usually have, okay, electrospray, ESI, or MALDI, which is matrix assisted laser desorption ionization. So if it depends on mass analyzer, and usually people are very picky about the mass analyzer, you can choose either quadruple if they have it, ion trap or ion trap quadruple, or time of flight. So usually ESI is coupled with quadruple or quadruple ion trap, and MALDI is coupled with time of flight. And there has never been ESI coupled with time of flight, okay, or MALDI coupled with quadruple or ion trap. So either source type, okay, can work with either analyzer, MALDI-TOF, okay, or ESI quad, and analyzers can be combined to create hybrid instrument. For example, ESI triple Q or MALDI QQ-TOF, okay, or Q-trap. Okay? So which means that, again, nowadays we have, okay, MALDI, okay, this is actually the latest one that they have developed, okay, MALDI QQ-TOF, okay, quad, okay, two quads, okay, but with time of flight, okay, and they basically now have, okay, MALDI Q-trap, okay, Q ion trap, but basically sensitivity is a problem, okay, for this, okay, for MALDI Q-trap, but they actually have the hybrid instrument nowadays. So ESI, okay, triple Q is quite common nowadays to increase the sensitivity of PMF. Now let's have a look at some of the examples of mass spectrometer available. So we have a Voyager D STR MALDI-TOF. So basically this is from German. Actually how you pronounce the name is Germanish, okay? I mean in terms of the German pronunciation, but I'm not going to pronounce it as such. So what happens, okay, is that basically it is a MALDI-TOF, okay, but okay, it has more or less a special detection and also a special kind of analysis packed with it, and this is actually very expensive, millions, okay, in terms of the cost. So we also have, okay, for example, okay, the multi QQ-TOF, okay, or Q star ESI QQ-TOF, okay. So basically this would represent, okay, both of them, okay, it's just that the mass analyzer, okay, would be different, okay. Now for the Q trap, okay, so usually it's a linear ion trap, okay, on a triple quadrupole. So we know that quadrupole has 4 metal rods, okay, so now you have triple quadrupole. So you have Q1, Q2, and Q3. So this is basically, okay, where it pulls, okay. So when it goes through Q1, okay, it's precursor ion selection. When it goes to the second, okay, quadrupole, it's for fragmentation, okay, to fragmentize different kinds of ions. And Q3 finally is to trap, okay, the ion. So only trap one, okay, will exit and gets to be detected by the detector. So the summary, okay, is that, okay, to acquire a mass spectrum, okay, you need ionization, you need a filtering, okay, which is in the form of mass analyzer, and of course you need to have a good detector to detect your ion, okay, to produce your mass spectrum. All right, so let's basically just, okay, have a look at mass resolution. So how does it affect, okay, your result? So the mentionless ratio of mass to charge ratio value of a peak is usually divided by its width at half maximum intensity. So what it means is that, okay, it would resolve different molecular species with similar but distinct masses. You probably think that they are the same, okay, in the machine, but usually they have distinct masses of their own. So high resolution is of course desirable for accurate mass measurement, but sometimes that could also mean that you need to have a robust kind of system. So in terms of mass accuracy, which is very important, okay, basically how close a measurement it is done or basically given to you, okay, to its true value, theoretical or exact. So usually it is expressed in ppm, and this is how the ppm is being calculated. So usually when you have the results, okay, your results, okay, will be in terms of theoretical, okay, versus expected mass, and you could compare with them, okay, side by side. So now this is where I promised you earlier, okay, whereby we have a tandem MS, okay, or MS/MS. So it is, or what a tandem MS is it is a consecutive stages of mass analysis to detect fragment ions to give it a structural information. So just to make, well, whether your life easier or to make your mind even mind-boggling, okay, usually we have up until 10 ms now, okay. So tandem or quad ms is quite common, uh, but we have up until 10 ms, right? So then you can imagine again how complex it's going to be. So we're not going to cover that, so don't worry, okay, we're only going to cover ms plus ms and compare them. So what happens is that in tandem MS, okay, from your ion source, okay, during the fragmentation, okay, you have mixture of ions, okay, this is the parent ion. So when it goes through, okay, the first mass spectrometry, okay, using the mass analyzer, okay, so you get single ion, which is your daughter ions. So when it goes through the second, okay, analyzer, so it basically will be fragmentized and will be detected by the detector. So this is more or less how an MS workflow, okay, will be done, or basically a rough representation of how it will be detected by the detector. So for your peptide mixture, okay, you have only masses to start with, okay, so it's all jumbled up, mixed together. Okay, so in the first MS, okay, one peptide selected for the MS, okay, and for the second one, okay, the masses of all pieces give an MS spectrum, which means a combination, possible combination that you could get out of them. So for peptide fragmentation, so usually, okay, you have, for example, okay, different forms of amino acids, okay, from the peptide, and you could actually determine, okay, what amino acids they are from MS. And of course you will need some, usually the analyzer or the peptide analysis software is embedded with it, but you also need, okay, to do some extra work, okay, to do some comparison with a standard, or basically with a known target protein and whatnot, because you need to more or less compare, okay, the mass of each amino acid. Now, to put it roughly or summarize MS versus, okay, tandem MS, so workflow is more or less similar. It's just that the results, okay, which are different. So you digest your protein. When you go through MS, okay, what you get, okay, is that each peak, okay, would represent, one peptide based on its mass, okay, mass to charge ratio. Okay, but okay, if you further it down, okay, to tandem mass, okay, what you get, okay, is that each peak, okay, would represent one amino acids. You still get mass to charge ratio, but okay, each peak, okay, would basically more or less signifies one amino acids, okay, so the distance between them. So now let's more or less recap back, okay, on what MS is. Okay, so the fundamental of MS, okay, is as such. So it have to have an ion source, okay, and of course the probe means that the inlet, okay, whereby you inject in your sample, your protein samples that have been digested. So there is an electron beam over here, okay, your ionization source, okay, ESI or basically MALDI, okay, and there is an accelerator plate, okay, in hand. So this is all in the machine, which of course you could see with our naked eyes, and the machine is actually not bent such as this, okay? Even though representation is bent, okay, but the machine is actually quite straight, okay. It's just that, okay, rough schematic representation is as such. So once, okay, it has been ionized, okay, so this is where, okay, your, can you see the dots, okay, here, which means that these are your peptides, okay. So initially they are in a mixture. So once it have been basically ionized, okay, with an electron beam, okay, so they now, okay, have their own charge ion. So okay, they will basically line up, not to say line up, okay, just a rough representation, meaning that the smallest one, okay, usually will travel faster, or usually doesn't deviate, okay, from the high vacuum. Okay, over here to over here is actually in a high vacuum. Okay, so once it travels, okay, into the rod, okay, and depending on different kinds of mass analyzer, so there is also a magnetic source. So ions that are too light, okay, usually would bend too much, which means that, okay, kind of like, because you are too light, okay, if there is a wind, then you may get swayed easily and whatnot. So they may go here or here, will be bounced off, okay, and in the end, probably, okay, they will not be detected, meaning that, okay, there is also a high chance for that to occur. So apart from that, ions that are too heavy also bend too little, which means that they actually are too heavy, okay, they couldn't walk straight, or they couldn't basically go through their target place. So what happens, okay, is that they usually sometimes will also not be detected by the detector. Okay, so once it has passed through the magnet, in the flight tube, only ions of the right mass can enter the detector. Okay? And of course, there is a detector slip over here, which means that those ions that pass through here, will be channeled to the detector. Detector, will record it and produce your mass spectrum, or spectra. So the principle is as such, sample entry, ionization, this is basically fragmentation of the ions, charge, to have a charge, and then detected by the detector. So fragmentation of M, M is actually molecular ions, so fragmentation of molecular ions will produce a radical and a cation, as such, so you basically have this, molecular ion from your protein mixture. So in the end, when it goes through fragmentation after ionization, you will get a radical and also a cation, radical, cation, or cation, radical, and so forth, only those two. Hence, you actually only get cations that are detected by MS. So what happens with an ion? Hold on to the question. So that's why, in MS application, only cations are detected, so the plus charge ions are detected, and you will get the masses of them being given by the detector. So in the end, more or less the summary is that how does a mass spectrometer work? By ion source, mass analyzer, fragmentation, and of course by the detector, which is represented by the mass spectrum to present your information. Okay, so we basically just refresh back or summarize all this in a block diagram. So mass spectrometry using a high vacuum system, it has an inlet, ion source, mass analyzer, detector, and also data system. So for the high vacuum system, usually it's using turbo molecular pumps, and when you have your sample introduced into it, so it is being injected into the inlet, whether by HPLC or flow injection or sample plate, if you are using MALDI. And it goes through your ion source, so you can have lots of ion source, either MALDI, ESI, FAB, SIMs, EI or CI. So it goes through, the mass analyzer, fragmented, and detected, and of course, you get your mass spectrum, being recorded by the data system. So what are the applications of mass spectrometry? So there are lots of applications, but these four applications are the ones that is the focus at the moment. Pharmaceutical analysis, which is in the bioavailability studies, drug metabolism studies, and also pharmacokinetics, characterization of potential drugs, which is more or less what a lot of researchers are doing as well, drug degradation product analysis, screening of drug candidates, the most common as well, and also identifying drug targets. In terms of biomolecule characterization, so it's usually to come up with proteins and peptides that could be used either for molecular or for medical purposes or for food industry, and you could also get oligonucleotides out of it. For environmental analysis, usually it is to be used on pesticides, on food or crops, soil and groundwater contamination, to more or less investigate to what extent the contamination is and how or what would the impact be. So the last one is actually on forensic analysis and clinical study. So do you know that actually for fingerprinting, the ones that you have learned from genomics part is actually kind of like coupled with mass spectrometry, because when you do DNA fingerprinting, you have to verify with mass spectrometry as well. So that's why we have forensic analysis over here. All right. So this is, I think, the second last slide. So we have again to cap back or summarize what we have learned on mass spectrometry. So we have different types of mass spectrometry. We have multi-TOF, used for molecular weight measurements or reaction monitoring if you have a target protein. ESIQ, which means quadruple, used for molecular weight measurement, HPLCMS, reaction monitoring, protein structural studies, and ESI or NSQTOF. This is actually very sensitive. Use a nanocolumn, used for molecular weight measurements, reaction monitoring, protein structural studies, peptide sequencing, nucleotide sequencing. You could also do this, but it will be very costly. Macromolecular structure determination, for metabolites, and could also be done due to the extended mass to charge ratio range. So this is the last slide for mass spectrometry. This is to represent the different kinds of mass spectrometer a machine available worldwide. So you can have lots of different kinds of machine. So we have multi-task over here. Okay, we have basically different kinds of analyzers. So we have over here ESI ion trap MS. So this is, we have a general LC-ESI MS, then the mass spectrometry, which is being used a lot. Okay, Agilent is the common provider for them. You also have, again, a general amino acid analyzer. Um, Not as good as the MS, okay, but basically could analyze your amino acid, okay, to a certain extent. Okay, so we start off, of course, okay, with Edmund sequencer, but not many people, okay, would like to use it, okay, because if you were to refer back to your notes, it actually is quite complex, okay, to be done. Apart from that, okay, you can basically couple it, okay, with radio imaging, mass spectrometry imaging. So this is by desorption analysis, okay, which means that you cut your sample, okay, using cryomicrotome, okay, and then, okay, you subject it to the radio imaging, and you can actually get your proteins to be detected in mass spectrum. Plus, you also get the results, okay, in terms of the sections of your sample that you have cut. So there you have it on mass spectrometry, the simplest that I could come up with, okay, and hopefully it would provide sufficient information for you guys.